Welcome to part 3 of our Chapter 2 voiceover lecture. In this video, we'll finish the last section, macromolecules. Part 3, proteins. The functions of the proteins include acting as enzymes, functioning as transporters, carrying out cell signaling, allowing cellular and body movements, providing structural rigidity, and there are tens to hundreds of thousands of different proteins with all sorts of different functions in our cells. Let's begin our discussion of proteins by talking about the building blocks or monomers of proteins, aka the amino acids. I will use the abbreviation a to mean amino acid from now on. Amino acids are attached to one another via dehydration synthesis reactions. There are 20 different unique amino acids. In this picture below, you can see a representation of a protein with many of the different amino acids attached together like a string of beads. This is a diagram of the structure of a single amino acid. It is generalized to represent any of the 20 amino acids. There are 5 parts that you should know. In the middle, we have the central or alpha carbon. Remember, carbon always makes 4 bonds, so there are 4 groups attached to it. To the left, we have the amino group made up of a nitrogen and 2 hydrogens. To the right, we have the carboxylic acid group made up of a carbon, which is double bonded to an oxygen, single bonded to a second oxygen, and that oxygen is bound to a hydrogen. Up above, we have the side chain or variable group represented with the letter R. There are 20 different side chains, one unique side chain for each of the 20 different amino acids. And then finally down below is simply a hydrogen atom. You don't need to be able to draw or memorize every atom of an amino acid, but you should know the 5 groups and how they are arranged. This is a table of all 20 amino acids and their structures. Again, you don't have to memorize the amino acids, and you do not have to memorize all their structures. But you should know the 3 different groups of amino acids, which are shown here. Up above are the nonpolar amino acids, also called the hydrophobic or water-fearing amino acids. In the middle, we have the polar or hydrophilic, water-loving amino acids. And then at the bottom, we have the electrically charged amino acids, which have an ionic charge on their side chain. You can see the electrically charged amino acids are divided into 2 groups, the acidic or negatively charged amino acids, and then there are 3 basic or positively charged amino acids. This is a table of the hydrophobic or nonpolar amino acids. The side chain or variable groups are highlighted in a yellow box. Notice the simplest amino acid, glycine. Its side chain is a single hydrogen atom. One characteristic all nonpolar amino acids share is that their side chains are mainly composed of hydrocarbons. Hydrocarbons, as the name implies, are compounds mostly composed of hydrogen and carbon. Like other hydrocarbons, nonpolar amino acids do not mix well with water. The other hydrophobic amino acids include alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. This is a table of the polar or hydrophilic amino acids. The side chains are highlighted in a green box. Like other hydrophilic compounds, polar amino acids are attracted to water. The polar amino acids include serine, threonine, cysteine, tyrosine, asparagine, and glutamine. This is a table of the charged amino acids. Notice there are 2 groups, the acidic and basic, or the negatively charged and positively charged amino acids. The acidic amino acids include aspartic acid and glutamic acid. Notice their side chains have a negative charge. The basic or positively charged amino acids include lysine, arginine, and histidine, and notice you'll see the plus or positive charge on the side chains of these amino acids. Remember, proteins are long chains of amino acids. Proteins are polymers and are built by polymerization, specifically dehydration synthesis. Here you can see a depiction of this process. The amino group of one amino acid is attached to the carboxyl group of another amino acid. The resulting covalent bond that joins these two amino acids is called a peptide bond. Proteins are broken down by hydrolysis. Once a protein is built, it can be described at 4 different levels of structure. The first level is known as primary structure. This diagram is a depiction of the primary structure of a protein. The primary structure of a protein is simply the order of amino acids that occur in that protein. For example, in this example, the first amino acid is glycine, the second proline, third threonine, fourth glycine again, fifth threonine, and so on. So just simply stating the amino acids in the order that they occur is the primary structure of a protein. The primary structure of a protein is incredibly important. It determines the shape and function of that protein. Hemoglobin is an example of a protein that biologists have studied and understand quite well. Hemoglobin is the protein in our red blood cells that carries oxygen. It is crucial that our cells build hemoglobin with the proper primary structure. In other words, they need to have the proper order of amino acids. People with sickle cell anemia have an altered primary structure in their hemoglobin. Specifically, the 6th amino acid should be glutamate. Instead, we find valine in its place. Normal hemoglobin proteins are shaped like globs, and normal red blood cells in which they're found have a biconcave shape. In other words, they have dimples on both sides. In sickle cell anemia, the altered hemoglobin sticks together in long fibers, and these fibers alter the shape of the red blood cells into a crescent moon or sickle shape. Hemoglobin in this state does not carry oxygen as well. A decreased ability to carry oxygen in the blood is called anemia. Hence, the disorder is called sickle cell anemia. You can see from this example how important it is that our cells get the primary structure of a protein correct. The next level of structure in a protein is called secondary structure. Secondary structure is defined as the folding of the amino acid chain. To understand secondary structure, once again imagine the beaded necklace. Each bead represents an amino acid, and the string holding it together are the peptide bonds. Now imagine taking that string and folding it up on itself. Many folds in different proteins are just random, but there are a few folds that we see again and again and are a nice orderly pattern. Specifically, the 2 types of secondary structure that you need to know are alpha helices, or coils. They are shaped kind of like an old telephone cord. An example of a protein that exists as a pure alpha helix, just one big long coil, is the keratin in our hair. The other type of secondary structure you see again is beta pleated sheets. They are like an accordion or a pleated skirt, folded back and forth on itself. An example of a protein that contains mostly beta pleated sheets would be spider silk. It gives it its stretch and strength. The third level of protein structure that you need to know is tertiary structure. The tertiary structure of a protein is its overall 3-dimensional shape. I remember it as the third level is the 3D shape. The tertiary structure of a protein is reinforced by several different intermolecular forces. The first one you should know is hydrogen bonds. Just like hydrogen bonds help hold water molecules together, hydrogen bonds help hold a protein in its three-dimensional shape. The next force is hydrophobic interactions. Because hydrophobic or nonpolar amino acids want to stay away from water as much as possible, these amino acids tend to fold inward or cluster in the middle of the proteins. The next one to know is the disulfide bridge. There are certain amino acids, particularly cysteine, that form strong covalent bonds with other cysteines. Two sulfur atoms are involved, hence the name disulfide bridge. And then finally, there are ionic bonds that hold proteins in their shape. These ionic bonds occur between the negatively charged and positively charged, or acidic and basic amino acids. This picture is a three-dimensional representation of a protein. Specifically, this is the protein aquaporin. Notice its shape. This shape is called a beta barrel. This is the tertiary structure of aquaporin. You don't need to memorize the specifics of aquaporin. This is an example to illustrate the three-dimensional shape of a specific protein. Finally, we're at the fourth level, or the quaternary structure of a protein. Quaternary structure is the number of polypeptide chains found in a protein. What does that mean? Let's go back to our example of a beaded necklace. Again, each bead is an amino acid, and the strings holding the beads together are peptide bonds. Poly means many, peptide is referring to peptide bonds. So when you have a string of amino acids linked together by many peptide bonds, you have a polypeptide. A single polypeptide isn't quite yet a protein. It needs to be folded, and some proteins need to be combined with other distinct polypeptide chains. In the example shown here, we have 3 individual polypeptides swirled together to form what's called a triple helix. This example is the protein collagen. The protein pictured here is hemoglobin. Notice hemoglobin has sort of a donut or circular shape. That would be the tertiary structure of hemoglobin. But its quaternary structure is 4, because it's made up of 4 polypeptide chains. This is chain number 1, number 2, 3, and 4. So hemoglobin's quaternary structure is 4. Collagen's quaternary structure was 3. Some proteins have a quaternary structure of simply 1, others more than 4. It just varies from protein to protein. The final shape of a protein can be altered by the environment. This is known as denaturation. Denaturation is a change in the normal shape of a protein due to environmental factors. These factors include pH, salinity, and temperature. In our cells, there are certain proteins that help other proteins fold into the proper shape. These are called chaperones. Think about taking a piece of meat and putting it on the stove and all the changes that you see. The same thing goes for an egg. What's happening is the proteins are being altered by a change in temperature. In other words, the proteins are being denatured. You take an egg, you put it on a frying pan, it goes from being clear, liquidy, and runny to being opaque, white, and solid. In egg whites, the protein albumin exists as little globs or globular proteins. When they're denatured, the proteins open up, and like lots of little strings, they get tangled up on each other and create one solid mass. Hence, we go from a liquid to a solid when we cook eggs. One of the most important functions of proteins is to act as enzymes. Enzymes are catalysts. A catalyst increases the rate of a chemical reaction and is not consumed, it's not used up in the chemical reaction. Enzymes are biological catalysts. Usually the name of an enzyme will end in ASE. Enzymes are usually proteins, but sometimes RNA can act as an enzyme. This diagram illustrates how enzymes work. The beige or tan-colored blob represents a protein enzyme, specifically the enzyme sucrase. Remember, enzymes usually end in the suffix ase, and the beginning of the enzyme name usually tells you the chemical that's either broken down or created by that enzyme. In this case, the sugar sucrose is being broken down by this enzyme. Enzymes have a binding site or an active site where the molecule will bind to that protein. In this case, sucrose binds to sucrase in this blue area right here. Once the molecule is bound to the enzyme will change shape. In the case of sucrase, the shape change causes the disaccharide to be broken into two individual monosaccharides, namely glucose and fructose. Then the enzyme releases those monosaccharides and returns back to its original shape, where it can once again break down another sucrose molecule. Some things to notice here: the enzyme is never used up in the reaction, it's not consumed. And the molecule that was being acted upon by the enzyme is known as the substrate. The molecule that's being made by the enzyme, the two monosaccharides, are known as products. And now the fourth and final macromolecule, nucleic acids. There are two nucleic acids, DNA and RNA. DNA carries the genes. RNA often acts as a messenger in our cells and sometimes can act as an enzyme, just like a protein. Nucleic acids are like carbohydrates and proteins in that they are polymers, long chains of monomers. The monomer of a nucleic acid is called a nucleotide. Nucleotides are composed of three parts. One part is a sugar shaped like a pentagon known as pentose. There are two pentoses, deoxyribose and ribose. Deoxyribose is found exclusively in DNA. In fact, the D in DNA stands for deoxyribose. The other sugar, ribose, is found exclusively in RNA. In fact, the R in RNA, as you might have guessed, stands for ribose. Notice in deoxyribose, there is an oxygen lacking that you can find in ribose. This removal of an oxygen is known as being deoxygenated, hence the name deoxy. The second component of a nucleotide is the base. Bases in nucleotides contain the element nitrogen and so are often called nitrogenous bases. There are 5 bases found in nucleic acids: cytosine, thymine, uracil, adenine, and guanine. Notice uracil is found exclusively in RNA, thymine is found exclusively in DNA, and the other 3 are found in both types of nucleic acids. There are 2 categories of nitrogenous bases, the pyrimidines and the purines. One mnemonic for remembering the purines is that the element silver has the chemical symbol Ag. So if you remember the phrase pure silver or pure Ag, the purines are adenine and guanine. And then the other 3 bases are the pyrimidines. The third and final component of a nucleotide is the phosphate. The formula for phosphate is PO43-, 1 phosphorus, 4 oxygens with a -3 charge. You do not need to memorize the formula for phosphate. This diagram shows a complete nucleotide with all 3 components. In blue is the phosphate group, the phosphorus in the middle and the 4 oxygens surrounding it. In purple is the pentose sugar, and in yellow is a nitrogenous base. The pentose sugar could be either ribose or deoxyribose, and the nitrogenous base could be one of the 5 bases we talked about previously. There is another nucleotide-based molecule that is not in DNA or RNA, and this is known as ATP. ATP stands for adenosine triphosphate. You may have heard of ATP before. It's the energy currency of the cell. This is a diagram of ATP. It's very similar to the other nucleotides. It contains the base adenine, here in green, the sugar ribose in purple, but instead of one single phosphate, it has 3 phosphates. Phosphates are negatively charged. When you have like charges, they repel each other. It takes a lot of energy to bind all of these phosphates together, and also when you release these phosphates, it releases a lot of energy. This is why ATP is a good energy currency. DNA is a polymer of nucleotides. The nucleotides come together in 2 strands that wind around each other. What do we call this shape? You guessed it, the double helix. The 2 strands are held together by something called base pairing. The bases on one strand pair with bases on the other. Bases are very choosy with who they will partner with. The base adenine, abbreviated simply as A, will only pair with thymine, abbreviated as T. The reverse is true. Thymine or T will only pair with adenine or A's. Also, guanine or G will only pair with cytosine or C. And again, the reverse is true. C's will only pair with G's. Within a single strand, the nucleotides are linked together so that the phosphate of one nucleotide is linked to the sugar of the neighboring nucleotide. This repeats, and the resulting structure is known as the sugar-phosphate backbone. There were several scientists involved in the discovery of the structure of DNA. One of those scientists was Rosalind Franklin. It was her laboratory that first took an X-ray diffraction photograph of crystallized DNA. The scientists Francis Crick and James Watson, together with the photograph from Rosalind Franklin's laboratory, helped put all this information together and discover the final structure of DNA. It was them who figured out that the overall shape was a double helix and that the bases A's and T's always come together and G's and C's come together. They also knew about the sugar-phosphate backbone. The drawing here on the left is the original drawing that Watson and Crick included in their published paper where they first described the structure of DNA. James Watson has since written a biography about the discovery of DNA called The Double Helix. It is worth noting that the DNA strands come together in opposite directions. This is known as the anti-parallel structure of DNA. So the nucleotides on one strand are oriented in one direction, and the nucleotides on the other strand are oriented in the opposite direction. Let's not forget about the other nucleic acid, RNA. The structure of RNA is a little less orderly. RNA usually exists as a single strand, as can be seen here in the computer-generated model on the left. This blue line represents the single strand of RNA. Sometimes RNA will have a slight twist, like on the left. Usually RNA is like a balled up string, like you see here on the right. The model on the left represents mRNA, and the picture on the right is representing ribosomal RNA. We'll talk about the different types of RNA in Chapter 3. You should know certain things about each of the macromolecules. You should know the names of the monomers for each macromolecule. For example, the monomer of a carbohydrate was a monosaccharide. You should know the components of these monomers when they've been mentioned. So the monomer of a nucleic acid is a nucleotide, and there were 3 components to a nucleotide: the sugar, the nitrogenous base, and the phosphate. You should know the name of the polymer for each of the macromolecules. So for example, the name of the polymer in regards to a protein was a polypeptide. The polymer with regards to a carbohydrate was the polysaccharide. And you should know a little bit about the process of polymerization or dehydration synthesis. When I've mentioned specific examples of macromolecules, you should know those. So you should know the 3 monosaccharides of carbs. Those would be glucose, fructose, and galactose. And you should know the functions of each macromolecule. For example, phospholipids function as the cell membrane or phospholipid bilayers that are found in our cells. And that concludes Chapter 2, the chemistry of life,